Hello, everyone. My name is Natasa Skoko. I am the head of the Biotechnology Development Unit at the ICGEB in Trieste, Italy. I will talk about development of biologics and transfer of know-how to industry about our ICGEB approach. ICGEB, one of the ICGEB instruments of actions, is technology transfer to industry for the production of biotherapeutics and diagnostics. I would like to say a few words about biotherapeutics, biologics, and biosimilars. Biologics or biopharmaceuticals are medicine produced from living organisms using biotechnology techniques. And we all know that they have revolutionized the treatment of different diseases like anemia, diabetes, autoimmune disease, and cancers in the last decade. They are one of the fastest growing segments of the pharmaceutical industry. Biosimilar is a biologic that contains a version of the active substance of an already authorized original biological medical product, so-called reference medical product, following loss of patent protection. Why similar and not identical? Due to because of the inherent complexity of these molecules and because of the complex manufacturing process. Even small changes or differences in manufacturing processes may have significant impact on the quality, purity, biological characteristics, and activity, clinical activity of the final product. So when we talk about biosimilar, we are talking about similarity to the reference medical product in terms of quality characteristics, biological activity, safety, and efficacy based on a comprehensive comparability exercise between biosimilar and reference product. When we look at the biosimilars on the market, first generation of biosimilars included interferons, filgrastim, erythropoietin, insulins, growth hormone. And the second generation of biosimilars are monoclonal antibodies or fusion proteins of antibody receptor moieties. Biosimilars are 35 to 65% cheaper than the originator drug. The trend in the price reduction with the launch of biosimilars resulted in the increase in usage, as you can see here. So not just that, as we said, is decrease in the price, but as you can see, the incredible increase in the usage. Doctors are more confident to prescribe this medicine for different treatments to the higher number of patients as the price decreases. This increase in usage was heavily driven by the availability of biosimilars as well as the other factors such as expanded indications. We at ICGEB in the Biotechnology Development Group, we developed our model for the development of technologies to produce biologics, biosimilars, and to transfer this know-how, this knowledge to the industry. Our mission is to strengthen capacities and competencies of the local partners to accelerate the product availability at the most affordable price. How? We decided that over last 25 years, having the group Biotechnology Development Group at the ICGEB in Trieste, we developed in-house expertise for production of active pharmaceutical ingredients of the biologics biosimilars. Then we developed the packages to transfer know-how to the industry with no exclusivity. We really work on a knowledge sharing capacities. Biosimilar development foundation for approval is built on a robust analytical data packages that demonstrate similarities between reference product and the biosimilar molecule, as we said. So the development of similar biopharmaceutical products involves a stepwise approach, starting with analytical processes with characterization and evaluation of quality attributes of the product and followed by non-clinical and clinical studies. Similarities to the reference medical products in terms of quality characteristics are based on a comprehensive comparability exercise that needs to be performed. And this includes different pharmacotoxical assessment, pharmacodynamics, in the non-clinical trials, and later on clinical development. So basically at ICGEB in our group, we focus our attention in the last 25 years on the first stage of every process for the biosimilar development, that is product development and comparative analysis. So basically we thought if we develop in-house knowledge, then we can save all our partners, industrial partners, first few years of the development, so then they can faster reach the market, or they dedicate their workforce and money and investment in the further down processes that we do not cover, that is process development, scale up, and non-clinical clinical studies and regulatory agency reviews. So we are very happy to say that at the Biotechnology Development Unit that was launched in 1995, that we have covered and developed many dozen of the technologies for the production of biosimilars such as interferons, GCSF, erythropoietin, pegylated interferons, pegylated GCSF, insulin technologies, growth hormone, and long-lasting insulins. So all the process of the production for the biologics and biosimilars start with the genetically modified cells and their growth in the bioreactors, fermenters, and then purification part and the quality control assessment. Finally, we finish all this process with the concentrated recombinant protein. So at the Biotechnology Development Unit at ICGEB, we can cover upstream and downstream processing. We work with the 3 different cell lines, bacteria, yeast, and mammalian cell. We have fermentation facility unit for microbes up to 30 liter and 2 liter for mammalian cell. We have a protein refolding capacity up to 50 liter and expertise in all kinds of chromatography for the pilot scale purification of biologics. So we are going up to multigram scale. So these small columns are the columns that usually are people are used to see in their R&D, in the research and development laboratories. That is few milliliter chromatography column where they can purify the micrograms or milligrams of protein. At the Biotechnology Development Unit at ICGEB, we work with a much higher scale. So we develop the expertise in the scaling up process. So we work even with the columns that has few liter of resin, and we can purify the protein in the multigram scale. So then it's easier for our partners, industrial partners, to scale up further from this pilot scale, pre-industrial scale to the industrial scale at their own facilities. We have developed very good synthetic organic chemistry laboratory where we can do peptide and protein chemistry, where we developed our own technologies for the polymer chemistry, polyethylene glycol chemistry, generation and activation, and we do all kinds of protein conjugation with chemical moieties, polymers, and fatty acids. Then last but not least, we are covering the quality control test following European pharmacopoeia guidelines. So we can perform all these kind of tests in our quality control laboratory. So I told you that we covered all 3 platforms, bacteria, yeast, and mammalian cell. And these are all the molecules that we have developed over the years. So the steps in technology transfer agreement and training in our group at the ICGEB starts as very simple and straightforward. And we start with finalization of the technology transfer agreement between ICGEB and the industrial partner through our technology transfer office. Then we offer up to 6 weeks training in the ICGEB laboratory in the Biotechnology Development Unit for our industrial partners to gain hands-on experience in the production. So we do at least 1 to 2 batches of the production, purification, and quality control for a certain biosimilar. And from 2020, due to the COVID pandemic, we developed also video-based training that you can find on this website. There are more information about this site. So we thought that even if there are travel restrictions, we want to be close to our industrial partners. And now we can offer some of these trainings in the video-based format. Then, after the training, we provide the protocols for the downstream, upstream, and quality control procedures. And if it's necessary, we assist the industrial partner in establishing the process in their own facility at this scale. Further on, we are very happy to say that we have more than 25 years experience in this field of production of biologics and biosimilars, development of biologics and biosimilars. We have signed more than 70 agreements with companies from 22 countries, and we train more than 100 scientists in our lab. As I told you, from 2020, we developed online training modules. Here in the world map, you can see that in the blue are countries where technologies have been transferred, and some countries in green are the technology that succeeded to reach market in their own country. We are very happy to say that a few years ago, we received a substantial grant from the 3 million euros from the region Friuli Venezia Giulia, where Trieste is based, to build a completely new biotechnology development unit, fully equipped laboratory comprising the spaces for the following process in the pharma-compliant environment, in the clean rooms class C and D. We have for the mammalian cell line development facility, upstream, downstream processes, polymer preparation, protein polymer conjugation process lab, and quality control lab for biologics and biosimilars. We are very happy to say that all the last year, in the record of 11 months, we succeeded to build all these laboratories in 215 square meters. And in green, you can see the labs, are the labs that are in the class D, and in the orange are the class C room, where we based our mammalian cell line facility. This is how all the laboratory equipment looks like in this new biotechnology development unit. And I would like to invite you to spend my last 3 minutes to show you the video how this dream came true. So how the building looked like when we received it in the March last year, and we succeeded to finish in the March this year. So please enjoy this video.
This is my last slide, and this is the Biotechnology Development Unit team. Very small team, but a team of very dedicated and hard workers. And I also included you here, our newly launched web page of Biotechnology Development Unit, where you can get more information about our technologies and our technology transfer model. And I also included here my email address if you want to contact me directly. I would like to thank you for your attention.